This is a very bad print, but this is the original from the study, where they looked at mortality from infectious disease in rural Gambia. And I think what you'll see, which is quite striking, is that there's this sharp increase in mortality during the first few years of life, but then almost completely leveled off from about the age of 5. And so there's clearly this period where you're at increased risk of mortality from infectious disease. And what we believe is that this doesn't just represent a lack of immunity, but it's actually because you are actually more poised to develop immune tolerance during this early window. And these were studies that were first established by Medullary in the 1950s. And he showed that you could introduce a foreign antigen in the embryonic or immediate postnatal period, and you could induce lasting tolerance to that antigen. And this came to be known as neonatal immune privilege. And he won the Nobel Prize for this discovery. But decades later, we really don't have a good understanding of kind of what the cellular mechanisms are that underlie this tolerogenic phenotype in neonates. And so you'll often hear people say, well, it's just because the immune system is not yet very well developed. And that kind of leads us to this idea that tolerance is just something that happens by default, that it's a passive phenomenon. And it's just because either the T cells are not very well developed to induce inflammatory immune responses, or the cells that are supposed to prime the T cells are not very effective. But what has sort of emerged over the past 10 years is that instead of this passive lack of immunity, is that immune tolerance is actually mediated in a dominant manner by these distinct subsets of T cells, regulatory T cells, or Treg cells for short. And these cells emerge in 2 waves. So first of all, you have Treg cells that develop within the thymus. And those Treg cells are specific for self-antigens. So these are Treg cells that are going to suppress unwanted responses to self-proteins that the T cells are going to encounter once they leave the thymus and then circulate within the periphery. But then you have another wave of regulatory T cells that we call peripheral Treg cells, or pTreg cells for short. And these are Treg cells that develop from naive T cells when they encounter harmless foreign antigens, such as microbial antigens or dietary antigens. And then they develop into these immunosuppressive regulatory T cells and suppress unwanted immune responses against those harmless proteins. And these peripheral Treg cells can be distinguished from their thymic counterparts by expression of this transcription factor, RORγt. And I'm going to talk a lot about RORγt later in the talk. And so a question then becomes is what induces these regulatory T cells, particularly in early life? And so for the thymic Treg cells, this is the pathway is very well established, and that these thymic Treg cells develop when this developing T cell encounters self-antigen. And it's actually a very beautiful story because obviously in the thymus, you don't express all of the cell proteins that you're going to encounter when you exit into the periphery. And so the thymus has come up with a very beautiful way to present cell peptides, and that's through expression of this molecule, air, or autoimmune regulator. And what air does is expressed by this unique population of thymic epithelial cells, and air turns on expression of genes in a promiscuous manner. And so these medullary thymic epithelial cells end up expressing a whole host of cell proteins that are not normally expressed by epithelial cells. And some classic examples of things like the insulin protein, which is normally only expressed in the pancreas, but now it will be expressed by this thymic epithelial cell. And so any T cell that recognizes these cell peptides will either undergo deletion or will get converted to this regulatory T cell. And so that's what happens in the thymus. So then the question is what happens in the periphery? So now a naive T cell is going to encounter these harmless antigens from the microbiota or from the diet. But what is going to instruct the differentiation of this naive T cell into this regulatory T cell? And in the lab, we're very focused on this cell, which is an antigen-presenting cell, which provides the cues to the naive T cell and tells it what to become. Now, we don't know very much, or we didn't know very much about the nature of the antigen-presenting cell that instructs naive T cells to become a regulatory T cell. But we do know that the window of encounter is very important. So this really kind of leads us back to this idea that the neonatal period is this kind of real window of opportunity, because several studies have shown that you effectively had this period which in mice exists around 2 to 3 weeks of age, where a naive T cell is far more likely to become a Treg cell if it encounters commensal or dietary antigens during this period. And so this sort of gave us a clue that there might be something unique about the antigen-presenting cell that exists during this early life period within the intestine. And so in the lab, we're very interested, as I mentioned, in studying these antigen-presenting cells. And the classical antigen-presenting cell is a dendritic cell. And we think of dendritic cells as really the sentinels of the immune system. So they sit within tissues and lymph nodes, and they're constantly sampling the environment, phagocytosing dying cells. And then they present antigens on their surface to T cells. And then obviously the T cells are what end up coordinating the overall immune response. And the T cell response obviously comes in many flavors. So you have CD4 and CD8 T cells. And especially within the CD4 T cell compartment, we have a huge amount of heterogeneity. So a naive CD4 T cell can differentiate into a number of different lineages. And each one of these different lineages is adapted for specific effector functions. But obviously, as well as these sort of inflammatory T cells that mediate responses against different microbes, you also have this immunosuppressive cell type that I mentioned. And so the classical view was one, where dendritic cells would direct these responses. And we became very interested in this idea that there might be a division of labor within antigen-presenting cells, whereby for each T cell phenotype that you want to elicit, there would be a different antigen-presenting cell that would already express the molecular programs required to induce these different differentiation programs within T cells. And so we became particularly interested in the identity of this antigen-presenting cell that might direct regulatory T cell differentiation. And when we started this work, sort of the classical view was that the balance between tolerance, for example, to microbes or dietary antigens versus the inflammatory response to pathogens would be mediated by dendritic cells. But instead of there being different subsets that would mediate this response, that the dendritic cell in its natural state, in the homeostasis, would exist in a tolerogenic state that could induce this immunosuppressive response, but that if it sensed invading pathogen, it would change its phenotype, undergo activation, and upregulate co-stimulatory molecules and cytokines. And then instead of eliciting regulatory T cell differentiation, would elicit an effective T cell differentiation. But I mentioned that we were more interested in this idea that maybe this was a compartmentalized phenomenon. And we had a clue that actually there might be a specific antigen-presenting cell that might regulate tolerance. And that came from earlier work from Greg Sonnenberg's lab, where he had shown that you needed antigen presentation by a cell that expressed this transcription factor, raw gamma T. And at the time when these studies were published, the only cell type that was thought to express both raw gamma T and MHC class 2, the antigen presenting molecule, was this group of innate immune cells called innate lymphoid cells. And one of the ILCs, the group 3 ILCs, expressed raw gamma T. And so the feeling at the time was that these ILC3s presented antigen and would regulate tolerance to the microbiota. But the only thing that was a bit unusual is that group 3 innate lymphoid cells had never been shown to actually directly present antigen to T cells. So it was kind of unclear how this tolerance was being mediated. And when I was a postdoc in Sasha Rudensky's lab, I had identified 2 distinct subsets of dendritic cells within the CDC2 lineage. And we had noticed that in one of the subsets, which we refer to as CDC2B, we were able to identify raw gamma transcript. And so we started to wonder whether actually it might be a raw gamma T expressing dendritic cell that was responsible for regulating tolerance to the microbiota. And so what we did is we returned to the mouse model where you delete antigen presentation on raw gamma T expressing cells. And this time we decided to look in mice at 3 weeks of age, because this is the window at which regulatory T cells are first emerging within the intestine. And interestingly, what we found is that these cells, although that they have normal levels of FOXP3 expressing regulatory T cells, they specifically lack the subset that express this transcription factor, raw gamma T. And these are the cells that represent the peripherally converted regulatory T cells that arise in the intestine that recognize your gut bacteria. And consistent with this loss of raw gamma T expressing Treg cells, the mice go on to develop this colitis, which is completely dependent on the presence of the microbiota. So at this point, we thought, okay, we now know that there is a raw gamma T expressing antigen presenting cell that is able to induce peripheral Treg generation. And so the question for us was, what was the identity of that cell? And so we decided to take an unbiased approach. And we generated a reporter mouse that enabled us to isolate all cells that expressed raw gamma. And we went into the lymph nodes in mice at 2 weeks of age, and we isolated all of the cells that expressed both raw gamma and MHC class 2. And then we performed single-cell RNA and attack sequencing. And this is the visual representation of that data. And what you can see is that the cells really break down into these different clusters, which are sort of colored separately. So these cells over here in green and blue, these represent the ILC3 cells, which were the kind of the classical raw gamma expressing antigen-presenting cells that were thought to exist. But then in addition to those cells, we also had this other group of cells that broke down into 4 distinct clusters. And so we became very intrigued by what these cells were. And when we looked at the gene expression profile for these cells, they didn't express any of the canonical markers for the ILC lineage. And instead, they had this very unusual profile. So they expressed genes related to epithelial cells, such as EpCAM, as well as TP63, which controls epithelial stem and progenitor cell differentiation. But they also expressed genes that are more classically associated with dendritic cells, such as FLIP3 and CBTP46, which at the time was thought to be a very specific dendritic cell marker. And so at this point, we did what all immunologists do when they don't know what they're looking at. And we turned to this ImGen database, which effectively has bulk RNA sequencing profiles of hundreds of different immune and stromal cells. And we just compared the transcriptome of our cells to all of the profiles from this database to see what cell type did our cells overlap with. And you can see here that when you look at the purple cells, which we had identified, they correlated with dendritic cells, particularly dendritic cells which express this factor CCR7, which represent the migratory dendritic cells which migrate from tissues to lymph nodes. But interestingly, they also had some correlation with medullary thymic epithelial cells, which are the cell type that I mentioned earlier, are very important in the thymus for generating thymic Treg cells. And when we looked more closely, we could see that the cells not only had some transcriptional overlap with MTEX, but they also expressed the marker which is used to define MTEX, which is the autoimmune regulator, which I told you has this very unique property in terms of being able to regulate self-antigen expression. And expression of air was actually limited to just 2 of the subsets. And so because of the hybrid phenotype of the cells, where they overlap both with dendritic cells and with epithelial cells, we named them Thetis cells after the Greek shape-shifting goddess, and we shortened that to TCs. And then the 4 subsets I just referred to as TC1 to 4. So I obviously showed you that these cells also overlap with dendritic cells. So one of the first questions we had is, well, are these just a variant of dendritic cells that acquire some properties of epithelial cells? And so to test this, we turned to this mouse model where you can label all of the progeny that come from this progenitor cell, this dendritic cell progenitor. So the dendritic cell progenitor expresses this gene CLEC9A, and so there is a mouse that exists, which is the CLEC9A Cree mouse, and so you can use that to label any cell that is descended from this CLEC9A progenitor. And so you can see here that when we read the CLEC9A fate mapper to our raw gamma reporter, you can see that we're able to label classical dendritic cells, but we don't label the TCs. And so what that tells us is that the TCs arise from a distinct progenitor, and I'll talk about that a bit later in the talk, that then gives rise to these 4 distinct subsets, which are separate from the dendritic cell lineage, even though there is some overlap in terms of their transcriptional phenotype. No, that's okay. I have a live mic for you. Okay, fantastic. Sorry, okay, hopefully everyone can hear me better now. And so, and then they adopt some features of dendritic cells, which we presume is because they have shared functions such as antigen presentation and migration. And so the immediate question for us was, well, what do these 4 different subsets do? And obviously the first thing we were interested in was their expression of air. And that was really because we know that air plays such a critical function in immune tolerance. So patients who have mutations in air develop widespread autoimmunity in childhood. And the mouse phenocopies that. So if you knock out air, you have, again, you have this very tissue-specific autoimmune disease. And so we wanted to understand more about what the function of air expression was in these cells and whether these cells might be contributing to tolerance in a manner similar to the epithelial cells. And so this is just showing again that they express not only the transcript, but they also express the air protein, specifically in this TC1 and TC3 subset. And so this was work that was led by 2 graduate students, Gaia in the lab, and then Zakia, who's a computational biology graduate student. And they really wanted to understand how much is shared between these two distinct cell types, the air-expressing MTECs and the air-expressing TCs. And so we performed parallel single-cell RNA and attack sequencing of these two different cell types. And this, I'm going to show you a bit more in detail. This is just the single-cell UMAP of the MTEC data, and then this is the TC data that I showed you previously. And now you can see that within the epithelial cell data within the thymus, we actually have quite a lot of heterogeneity. But in the middle, in this yellow cluster, this is where the air-expressing cells sit. Now when we perform a computational analysis called topic modeling, and what that does is that allows us to pull out shared gene expression programs, which are shared across cell types, even though they may not cluster together by cell identity. And so you can see here what we found is that there's a module of genes, which is this green topic here, that was specifically enriched within the air-expressing cells. And when we applied the same topic model to the TC dataset, we could find that the same module of genes was specifically enriched within the air-expressing TC1 subset. And so that suggests that these two cell types, the air-expressing MTECs and the air-expressing TC1s, have some common transcriptional properties that we think is going to represent a kind of shared module of tolerance, which we're currently investigating. Our current model is that the TC1 subset may be mediating tolerance to self-antigens in a manner similar to the thymic epithelial cells. But this suggests a role more for the cells in tolerance to self. And here, air actually turns out to be a bit of a red herring for the story that I started with, which is that we needed these cells to present antigen to tolerize against the microbiota. And what we found is that if you delete air from the cells, it actually has no impact on peripheral Treg generation. So in contrast to the role for air in the thymus, where you need air for thymic Treg generation, you don't need air expression in these cells to generate peripheral Treg cells. And so that was initially a little bit disappointing. And then we thought about this a little bit more, and we thought, well, actually, there's 4 subsets of cells here. And interestingly, when we started looking at the gene expression profiles of these cells, the one that really leapt out to us was this subset, TC4. And that was because TC4 was actually enriched in some key molecules that we know to be important for peripheral Treg generation. And when we performed the same topic modeling approach, we pulled out this module of genes that we refer to as this Treg inducing module. And that's because it encompasses these molecules that have been shown previously to be absolutely critical for naive T cells to undergo peripheral Treg generation. And the most important one is this one, integrin beta 8. And so integrin beta 8 forms part of this complex with integrin alpha V. And then you need integrin beta 8 to activate latent TGF beta. And TGF beta is the key cytokine that allows a naive T cell to become a peripheral Treg cell. And so we started looking at expression of integrin beta 8 in more detail. And what you can see is that amongst the raw gamma T expressing cells, it was specifically expressed by the TCs, but not by the ILC3 subsets. And so we thought, okay, we could confirm this with an integrin beta 8 reporter mouse. You can see here that we see expression within, particularly within the TC4 subset, but not within the ILC3s. And so we made a mouse where we can delete integrin beta 8 within raw gamma T expressing cells to see what would happen to peripheral Treg generation within the intestine. And you can see here that this mouse completely phenocopies the raw gamma T Cre class 2 flox mouse. So you now lose peripheral Treg cells within the gut lymph nodes and the large intestine. And again, these mice go on to develop a microbiota dependent colitis. And so what that tells you is that you need both antigen presentation and integrin beta 8 expression by this TC4 subset to induce peripheral Treg generation. Now, I told you at the beginning that before we started this work, the kind of conventional view was that ILC3s were the antigen presenting cell that was performing this function. And we were pretty sure because of the integrin beta 8 expression, specifically within TCs, that this was a TC mediated phenomena. But we went back to our data to see if we could generate mouse models that would be specific for targeting either ILC3s or TCs. And we found within our gene expression data that there were a number of genes that distinguished ILC3s, including this gene, raw alpha. And that was very interesting to us because there was already a raw alpha Cre mouse. And so we thought, okay, we can use this mouse to see if we can target ILC3s. But there was one caveat to this, which is that the ILC progenitor expresses raw alpha. And so if TCs were descended from the ILC progenitor, then they would also be targeted by this Cre. So we first of all bred the raw alpha to a fate mapping mouse. And then we could look within TCs and ILC3s to see which of the subsets were labeled by raw alpha. And you can see here again that the ILC3s are labeled by the raw alpha Cree, but we don't see labeling within the TC subset. And so we could move forward with this mouse, and we deleted MHC2 on raw alpha expressing cells. And I'm just showing you here, you can see that we have this loss of MHC2 on ILC3s, but the TCs are spared. And when you look at the intestine of these mice, they have a completely normal intestine. So they have normal numbers of regulatory T cells and Th17 cells, and histologically, the guts are completely normal. So at this point, we knew that amongst the raw gamma expressing cells, it had to be the TCs that were responsible for mediating tolerance to the microbiota. And this is obviously also very different from, the classical view, which is where dendritic cells mediate tolerance. And so we wanted to be completely sure that this wasn't a dendritic cell, that dendritic cells had no role in this process. And so we now knew that we could distinguish TCs and dendritic cells using this CLEC9A Cree mouse that I mentioned previously. And so we generated a mouse where we deleted class 2 expression on dendritic cells. And we also found that although we lost class 2 expression on these cells, we didn't have any impact on the peripheral Treg cells within the intestine. So again, suggesting that the dendritic cells were also dispensable for tolerance within the intestine. So I started this talk by saying that we thought that there was this developmental window for immune tolerance and tolerance to the microbiota. So the question is how do TCs regulate that developmental window? And so we know that the peripheral Treg cells start to emerge at this time point, which is around 2 weeks of age. And this is a very interesting window in mice because this is when mice start to wean from milk to solid food. And then there's a huge bloom in the microbiota. And this is the time point at which you start to get regulatory T cells in response to your gut microbiota. So we looked at the numbers of TCs within the gut draining lymph nodes, which is where peripheral Treg generation happens. And one of the things that was just really striking to us was that the cells dramatically increase between day 7 and day 14, but then they come back down again. And so from about 3 weeks of age, you have very low numbers of TCs within the gut draining lymph nodes. And so we thought, okay, well, maybe this explains why you have this window of opportunity for peripheral Treg generation, because these cells decline to very low numbers. And interestingly, this isn't just a mouse phenomenon. So we were also able to use publicly available data from the human cell atlas. And we were able to find a population of cells within that data set that resembled TCs. And they were also enriched within the fetal gut lymph nodes relative to the adult gut lymph nodes. So suggesting this phenomena is conserved across species. And so we think that TCs really regulate this developmental window for developing tolerance. And this is actually a relatively safe time to lock in tolerance by having this overabundance of tolerogenic cells, because at this time point, the developing infant actually has passive immunity to pathogens through transfer of maternal antibodies, which then actually decline with age. So in humans, after about 6 months, you have this decline in maternal antibodies, and that's obviously when infants start to get all of their infections. So we think that what happens is that the immune system effectively exists in this tolerogenic state by having this overabundance of cells which are going to induce tolerance to harmless foreign antigens such as commensal antigens. But of course, this is not the only time window which you need to regulate tolerance. So we know that later in life, you might have incidences of inflammation, and inflammation is often a trigger for autoimmunity. And what's interesting is that we found that under certain conditions, the TCs will expand back up again. So I'm showing you here in a mouse model of CNS autoimmunity. This is experimental autoimmune encephalitis, which is really a mouse model for MS. And you can see here that the TCs increase within the draining lymph node following induction of disease, and then they come back down again. And interestingly, we also have looked in tumor settings, and after tumor induction of tumors, this is a subcutaneous tumor that's been implanted, we find that the TCs again expand up within the tumor draining lymph node, not just within young, but also with adult mice. So we think that anytime that you have these settings where you need to maintain tolerance, or perhaps a tumor has co-opted a pathway of physiological tolerance, that these cells will expand back up. So our current thinking is that you have these 4 distinct subsets. We think that TC1 is going to end up playing a role in tolerance to self, and that's work that's currently ongoing. And then now we've shown that TC4 regulates tolerance to the gut microbiota. But the gut microbiota is not the only thing that you have to regulate tolerance to, particularly within early life. So we started wondering, well, do TCs also play a role in tolerance to other foreign antigens? And of course, across all of these different organs, each tissue is faced with its own unique challenges in terms of what it has to regulate tolerance to. But the other key property of the intestine is that it has to be able to induce tolerance to your dietary antigens that you ingest. And again, what was very interesting to us is that this time window then becomes very interesting because the TCs peak just at the point that you're weaning from milk to solid food. And this is when you're going to really encounter your dietary antigens for the first time. And particularly within mice, this happens during a very narrow time window between 12 to 18 days. And we know actually now from studies in humans that the early life window represents this real window of opportunity for establishing tolerance to food antigens. And so this is from a clinical study that was actually performed in the UK. And the UK, there was advice at the time that had come out actually decades earlier, where they, in response to the increasing peanut allergies, the advice from the government had been to actually delay onset of peanuts in high-risk individuals. And so the guidance at the time was that children shouldn't start eating peanuts until they're about 5 years of age. And interestingly, what had happened is that peanut allergies, the incidence had started kind of like increasing like this. And so one of the physician scientists who was in the lab next to me where I was doing my PhD, Gideon Lack, actually had thought, well, maybe what's happening is that you're missing the natural window in which you can induce tolerance to these antigens. So he conducted this clinical trial where he showed that if you introduced peanut allergens early into high-risk individuals, so these were infants that had a sibling that already had a past history of peanut allergy, you can see here that you have this dramatic reduction in peanut allergy in the consumption group. So these are the infants that are actually exposed to peanut through like peanut butter. And so this really, I think, is a very interesting human study where it highlights the fact that this window of opportunity is not just exists in mice, but it's also common to humans. And currently the field, in terms of the food allergy field, is really moving in the direction of early introduction of allergens. And if you go to the conferences now, the discussion is how early is too early? Like how early can you start exposing developing neonates to different types of food, which we know can also happen through passage through the breast milk. So we started thinking, okay, let's think more broadly about what the role of TCs might be in tolerance, not just to the microbiota. And so previously we had really focused on these cells in the context of the large intestine because that's the predominant site of tolerance to the microbiota. So we decided to look in all of the different gut draining lymph nodes. So here you have D1 and D2 represent your duodenal lymph nodes, and then you have your ileum, and then your jejunal, and then your colonic lymph nodes, and then also your Peyer's patches and your small and large intestine. And I think what you can see is that across all of the different lymph nodes, you have this complete loss of raw gamma T expressing Treg cells, which also represent the Treg cells that you develop in response to dietary antigens. So we thought, okay, and interestingly, what we also found is that if you age these mice, they also develop this inflammation within the small intestine. And so, and you can see this on the histology as well, where you have this goblet cell hyperplasia and lymphocytic infiltration. So 3 members of the lab decided to team up on this project to really ask the question, which is, do TCs also play a role in regulating tolerance to dietary antigens? And so this was a fantastic team effort. And they started just to ask the question, which is, okay, if you feed a mouse a egg protein, which is ovalbumin, which cells actually take it up? So which are the antigen-presenting cells that can present ovalbumin to the T cell? And so they use fluorescently labeled ovalbumin, and then you can see that there were really only 2 cell types that take up this protein. One is these CCR7 expressing dendritic cells, which I mentioned previously, which are your classical dendritic cells, which migrate from tissues to lymph nodes. And then we had uptake within the TC4 subset. So we became very interested in this and what the different roles might be of dendritic cells versus TCs in the context of tolerance to the dietary proteins. And so now what we did is we took our mouse model where we delete antigen presentation on raw gamma T expressing cells, and then we feed the mice ovalbumin. But we start feeding the mice at P14. So this is just at the point that they're weaning from milk to solid food. So we give them 2 gavages of ovalbumin, and then we leave the ovalbumin in the drinking water. And then 7 days later, we look to see what happens to this population of T cells that we transfer in that specifically recognizes the ovalbumin protein. And what we find is that you can identify these cells here based on congenic markers as well as their T cell receptor.
And you can see that in the wild type mice, these cells differentiate into Treg cells. They express FOXP3. Some of them express this, the transcription factor of raw gamma T. But in the mice that lack antigen presentation on raw gamma T expressing cells, we have this complete absence of regulatory T cell differentiation in response to ovalbumin feeding. And as a consequence of that, we can now see, well, what happens? Do these mice develop sensitization and allergy? And so in this model, what happens is you pre-feed mice with ovalbumin to induce oral tolerance. And then you try and come in and you see what happens if you try and break tolerance to ovalbumin by injecting the ovalbumin protein along with this adjuvant. So it's conjugated to alum. And then you expose them to ovalbumin through the nasal epithelia. And then we take the bronchoalveolar lavage fluid and we look for evidence of allergic inflammation by looking at the levels of eosinophils. And what you can see here is that here in the control mice, these are mice that didn't receive ovalbumin, so they have a slightly higher levels of eosinophils when they get exposed via the lungs. And then in the gray here, we have the mice that have expression of class 2. And you can see here that these mice are protected from eosinophilia, so they have very, very low levels. But in the mice where we've deleted class 2 on raw gamma T expressing cells, then you can see that they start to break tolerance to ovalbumin, and they actually have even higher levels of eosinophilia than the control mice. And so that suggested that actually, yes, it is TCs that might be regulating tolerance to both dietary antigens as well as the microbiota. Now, the nice thing about this system is that because we can introduce the antigen at a particular time period, we can now start to see, well, okay, what happens if we introduce the allergen outside of this normal window that we're saying is like the window for inducing tolerance? Because we know that you still have TCs at this point, but they're very low within the lymph nodes. So the question is well, what would happen if you ate something new outside this early life window? Would you not be able to tolerize to it? And so what we did to address this is we used a mouse model where we can temporarily delete class 2 expression on raw gamma cells using this tamoxifen-inducible Cree. And so we take adult mice, we put them on the tamoxifen diet for 2 weeks to ablate the class 2 expression. And then we introduce the ovalbumin into the diet. And then we see what happens to the OT2 cells that we transfer in. And actually, surprisingly, you can see that the wild type cells still develop some degree of tolerance. So we do get some Treg conversion within the lymph nodes and the small intestine. But the mice in which we've ablated class 2 on the raw gamma T expressing cells have no Treg cells. So suggesting that you still need TCs within adulthood if you're going to induce tolerance to these antigens. But the clue becomes is that when you compare the level of Treg induction in the adult mice versus the young mice, you can see that in the adult mice, we're actually only getting about 10 to 15% conversion within the lymph nodes. In contrast, when we did this experiment at 2 weeks of age, we were getting about 50 to 60% of the cells becoming Treg cells. And so that really seems to be the major difference between adults and neonates, is that TCs play a role in both, but that you will generate a lot more Treg cells during the neonatal period because that's really when you have the most amount of cells. And so it's much safer to try and establish tolerance then because you're going to generate a lot more of these immunosuppressive Treg cells. So then we obviously wanted to know, well, is it the same mechanism that by which TCs are regulating tolerance? And I showed you previously that they needed expression of integrin beta-8 to regulate tolerance to the microbiota. And so we wanted to know, would that be the same mechanism by which you would generate Treg cells to dietary antigens? And so this time we decided to isolate TCs from lots of different lymph nodes to see what the proportion was of these different subsets. And interestingly, what we found is that along the bottom here, the first four groups I'm showing you are different gut lymph nodes. So we've got colonic lymph nodes, and then the small intestinal lymph nodes, the hepatic lymph nodes, and then the Peyer's patches within the small intestine. And then we move on to the lung, and then we have the skin, the salivary lymph nodes, and then these para-aortic lymph nodes. And I think you can see that the pattern that emerges is that these dark cells here, which are the TC4 subset, are really enriched within the gut lymph nodes and then decline to very low levels kind of outside of the intestine. And it's really this TC4 subset, which is where you have these integrin beta-8 expressing cells. And we could confirm that with our integrin beta-8 reporter mouse, is that when we looked at the frequency of integrin beta-8 expressing cells, they were actually highest within the small intestinal lymph nodes and then the colonic lymph nodes, but you don't find any TC4 within the skin draining lymph nodes. And so we think that actually anywhere where you have a kind of pressure on tolerance, that there's some factor that is being produced that is actually inducing the differentiation of TC4s. And that's kind of work that is ongoing, and I'll talk a little bit about towards the end. So we went ahead and we deleted with our mouse, where we delete integrin beta 8 expression on raw gamma T expressing cells, and you can see that we completely lose these dietary induced Treg cells within the gut lymph nodes. And so to just really sort of nail this and start to nail it down to like one particular cell type and prove that there was no redundancy between different antigen presenting cells, we made a mixed bone marrow chimera setting where the mice receives either bone marrow from mice that lack the MHC class 2 expression on raw gamma T expressing cells, or they lack integrin beta 8 expression on raw gamma T expressing cells. And so in this setting, you have cells that can present raw gamma T expressing cells that can present antigen, or you have raw gamma T expressing cells that can activate TGF beta, but no cell can do the same job. And interestingly, what we find in that setting is that we have our various controls in gray, and then we have mice that have just received bone marrow from the MHC raw gamma T expressing mouse, and you can see that they lack peripheral Treg induction, and so do the mice that receive the bone marrow just from the integrin beta 8 knockout mouse. But when you try and rescue the phenomenon by having half-half, you can't. And so what that tells you is you actually need TGF beta activation and MHC class 2 expression on the same cell to be able to induce Treg cells. And so what that suggests is that you have a very spatially controlled process where you can only activate TGF beta in this very local immunological synapse to get very tight control of these regulatory T cells that are going to develop. So really at this point, the major questions for us was, well, how is this whole pathway for TCs regulated, and what regulates their wave in early life, and then what regulates their expansion later during inflammation? Because we feel that if we can understand these cues, then these cells become a very interesting therapeutic target in settings of intestinal inflammatory disease, or for example, in food allergy. And so this was the question that we really wanted to address, is sort of what determines this wave of TCs in early life. And this is work that's led by a graduate student in the lab, Jocelyn. And she really wanted to understand, first of all, where do TCs come from, but also then what regulates their differentiation into these 4 distinct subsets. And obviously, I mentioned to you that we think that the tissue microenvironment plays a major role in this because of this loss of TC4s that we see in lymph nodes outside of the intestine. So she became very interested in sort of what was unique about the tissue environment within the intestine that might regulate the differentiation of these cells. And in the postnatal period, this is a kind of absolutely fascinating time within the intestine because, first of all, you're exposed to breast milk. And breast milk contains a whole host of immune modulatory factors. So it's enriched for cytokines such as TGF beta and EGF, which has been shown to regulate gut permeability. And then at the same time, the lymph nodes are still developing within mice. So you have this very unique cellular composition of different immune and non-immune cell types that are interacting with each other. And then, of course, as you wean from milk to solid food, your microbiota takes off, and then you have all of the metabolites that are being produced by the microbiota that have also been shown to have immune modulatory actions. And this is obviously a vast topic. So I'm just going to show you one aspect of this story, which is really a story about the cues from the developing lymph nodes that actually regulate TC differentiation. And so one of the things that Jocelyn did is she turned to our original data set where we had done single cell RNA, but also attack sequencing. And with the attack sequencing data, we can start to look at the chromatin accessibility between the different cell types. And that gives you a clue about which factors might be regulating the differentiation of these cells, because you can look for the transcription factor motifs that are present within the differentially accessible peaks. And what she found was that within the TCs, there was enrichment of motifs for these NF-kappa B signaling factors, which are REL and REL-B. And this was very interesting to us because these factors have been shown to play a very critical role in regulation of thymic epithelial cell differentiation. And I mentioned that these 2 cell types have kind of like interesting transcriptional parallels. And so we already knew that within the thymus that the differentiation of the air-expressing MTEX was dependent on non-canonical NF-kappa B signaling. And upstream of the NF-kappa B and MTEX is this molecule called rank ligand, which has been shown to play a very critical role in MTEC differentiation. And so we went back to our RNA sequencing data set, and we started looking for expression of molecules within the NF-kappa B pathway to see which molecules might be regulating TC differentiation. And interestingly, we were able to show that the receptor for rank ligand was expressed by some of the TC subsets. This is this molecule here, TNF-RSF11A. And so this is the molecule that she went after first, although there's actually a whole family of them. And this, and you'll see later that this turns out to be very subset specific. So we think different NF-kappa B signaling molecules might be regulating each of the different subsets. So first of all, she used this rank reporter mice to see whether the different TC subsets did indeed express rank. And what she could find is that she found highest expression within TC1s, and then the expression kind of declines from the TC1, and then TC4s have very little expression of rank. And this turns out to be very important when we look at the phenotypes of the mice that develop when you block rank ligand signaling. So first of all, she took a pharmacological approach, and she gave anti-rank ligand blocking antibody. And she gave that antibody to mice at day 7 of age, and then continued the treatment until they were 2 weeks of age. And what she could show is that she completely lost this TC1 subset that we can distinguish through expression of this molecule NCAM1. And this is just showing a summary for the different TC subsets. You can see we have a complete loss of TC1, and then a partial loss of TC2, and then TC3 and TC4 are spared. And that really fits with the expression pattern of the rank receptor that I showed previously. So then we wanted to take a genetic approach to really selectively target rank expression within the RORγt expressing cells. And so we deleted the receptor using the RORγt CRE, and then we analyzed these mice at 16 days of age. And again, she found exactly the same thing, that you completely lose this TC1 subset. And you can just see that here. Again, we have the same pattern as before, this loss of TC1s and then this partial loss of TC2s. So at this point, we started thinking, okay, well, which cell is making the rank ligand, which is regulating availability of this molecule for TCs? And so she turned to the human cell atlas data that I mentioned previously, where they had profiled all cells within the developing intestine and the lymph nodes. And we thought, okay, well, this is probably the best data set. We can go in an unbiased manner and see who's expressing rank ligand. And what you can see here in the lymph nodes is that the major cell type that's expressing rank ligand is ILC3s. And then this interesting cell, which actually expresses much higher levels, which are these lymphoid tissue organizer cells. And these cells, you can see that they're really enriched in the embryonic period within humans. And so just a little bit of background about LTO cells. So lymphoid tissue organizer cells act in concert with another cell type, LTI cells, which are lymphoid tissue inducer cells, which are a variant of ILC3s. And these two cell types are effectively the two cell types that are required to coordinate lymphoid tissue formation during early life. So the LTI cells seed the sites of lymph node tissue formation first, and then they produce lymphotoxin beta, which acts on LTO cells, and then LTO cells produce more rank ligand, which acts on LTI cells, and then there's this feedback loop that happens, and you get kind of successive differentiation of these cells, and then recruitment of other immune cells to the developing lymph node. And this sort of process happens in sequence. And so we thought, well, this is very interesting, because this is therefore a way of coordinating TC differentiation with lymph node tissue development to make sure that the TCs develop as the lymph node tissue is forming, so that you have these tolerogenic cells that are present right at the beginning of lymphoid tissue formation. And so we wondered whether this rank ligand production by either LTI cells or the LTO cells promoted TC differentiation. So I'm not going to show you the negative data. It turned out that LTI cells were dispensable for this process. But when we looked at production of rank ligand by the LTO cells, we used a genetic mouse model where we deleted rank ligand by these cells using this twist2 CRE, which is expressed by LTO cells. And then this mouse, again, almost completely phenocopied the mouse I showed you earlier, where we again lose TC1s. And you can see that here, that we have this loss of TC1s within the developing lymph nodes. And this is very interesting, because LTO cells have already been shown to, first of all, be enriched within gut-draining lymph nodes relative to skin-draining lymph nodes. But also there was a previous paper that showed that they were also enriched during early life. So LTO cells themselves almost have a clock, whereby they come in and they help form lymph nodes in early life, but then they disappear. And so this window of these cells for producing rank ligand is again temporally restricted. And so we think that this may explain the temporal restriction that we see, at least for the TC1 and TC2 subsets. And we're now exploring whether there are other factors within the NF-kappa B signaling pathway that regulate the TC3 and TC4 subset. And we think that converging on this NF-kappa B signaling pathway are signals that you receive from both the breast milk and the microbiota. So I'm going to end there and just kind of present our sort of current summary for how we think immune tolerance is regulated. So the classical view before we started was that thymic tolerance was mediated through this specialized lineage of epithelial cells that express the autoimmune regulator, but that within the periphery, tolerance was actually regulated by dendritic cells that could switch between this tolerogenic or pro-inflammatory phenotype. But we now think that there's actually a division of labor so that you have a dedicated lineage of tolerogenic antigen-presenting cells, which actually have interesting transcriptional overlaps with medullary thymic epithelial cells. And that now you will have this division of labor so that you either have pro-inflammatory dendritic cells or you have tolerogenic Thetis cells. And obviously the key questions for us now are establishing the roles for the different TC subsets in different aspects of immune tolerance. So I've shown you that TC4 regulates tolerance to microbial and dietary antigens, but of course there's a whole host of other foreign antigens, and we're very obviously interested in the role of these cells, for example, in tolerance to neoantigens in the context of tumor formation, and also their roles in self-tolerance. So I'm going to end there and actually do the most important part, which is acknowledging all of the people who've done the work. I've sort of shown you their faces as we've gone along, but they're really a fantastic team of scientists, and it wouldn't have been possible to do any of this work without them. And we've had wonderful collaborators along the way, almost too many to mention, but I think really the key collaborators that need acknowledging are the computational biologists who really have done exceptional amounts of work on these cell types. And then, of course, all of our funders. And so I'm going to end there and take any questions on the scientific part. But I'm also very, very happy afterwards to kind of really talk about my scientific journey and how I got to where I got to, and happy to take any questions on that as well.
Start. What's your sense on the importance of microbial diversity in triggering tolerance? Is population size and density more important? Is a diverse community more important, or both?
Yeah, it's interesting. So there was a study that came out a while ago from Christophe Benoit's lab, where when the raw gamma T positive Treg cells were first characterized, they did these very nice studies where they obviously showed that germ-free mice don't have any. And then they started introducing different microbes to see kind of which would be the ones that could induce peripheral Treg cells. Now, interestingly, sometimes all you need to do is introduce like one particular strain, and you can get the peripheral Treg cells back up to the levels of, you know, a dirty kind of SDF mouse. And then, of course, you have other strains that have kind of moderate phenotypes, and then some that can't induce peripheral Treg cells at all, which I think is really something the field is focused on, which is what is it that's unique about kind of those cells that are high P-Treg inducers. And some of it might be about, you know, how their antigens are accessed, where they colonize within the intestine, whether they can be captured by the antigen-presenting cells and seen by the T cells. But I think the other thing that is also now becomes apparent is that in a very recent study that came out, it was shown that if you look at the T cell specificity of peripheral Treg cells that recognize the microbiota, it's actually incredibly promiscuous. So unlike what happens in other settings where we're taught that, you know, the T cells have incredible specificity to recognize these specific antigens, and that's how you can tailor T cell responses to one pathogen versus another, what seems to happen with the microbiota is that you actually generate tolerance to very conserved antigens that are conserved across quite diverse strains of microbiota. And so maybe the idea is there that actually you lock in tolerance to the microbiota that you've seen during early life. And even though you might encounter new microbiota, you're going to end up tolerizing it against the same antigens that are conserved across the different strains.
Thank you.
These Thetis cells are really fascinating. And so I'm wondering, is it possible to upregulate them, overregulate them, like overproduce them? And do you see a greater susceptibility to infection if you do? Since you're basically potentially creating more tolerance.
Yeah, I think that's a great question. And that's definitely what we're trying to understand at the moment, which is how much can we manipulate their levels outside of the normal setting? I've obviously shown you that different inflammatory settings, we can see this increase in TCs, but I would say we still don't yet have a very good handle on kind of what is the exact cue that the cells are sensing that would allow us to go in and kind of manipulate their levels therapeutically. So for example, in the rank ligand story, where we now, at least for the TC1 subset, have really kind of delineated kind of what the signaling pathway is that regulates their differentiation, we can enhance their differentiation potentially through through expression of this, through deleting this molecule called OPG. OPG is the decoy receptor for rank ligand. So if you delete the decoy receptor, then you effectively have more rank ligand signaling. So those are experiments that we're doing at the moment to see what happens if you knock that out.
I guess we're not all equal in terms of how many of these TCs we retain when we age. Do you see a difference, a variation in the normal population and in people, let's say, that are maybe either immunosuppressed or have chronic inflammation or things like that?
Yeah, I think that's a great question. So one of the challenges for us is that the cells exist within lymph nodes and not really within tissues. And of course, that's the hardest site for us to access for human specimens, because when you have disease, you normally biopsy the tissue, but you very rarely get lymph nodes. So we're just at the moment establishing a collaboration with Donna Faber's group, because she has a collaboration with the transplant organizations where they get tissue and lymph nodes from deceased donors. And so she's able to profile human cells within lymph nodes across different ages. And she's had some beautiful studies on that in relation to T cell development. And so now the idea is we're going to go back and ask exactly that same question in the context of TCs, which is how do they vary with age and how much do they vary from one individual to another? Yeah.
So you showed this really interesting TC spike.
Yeah.
As the infants are weaned and start taking on solid food.
Yeah.
You showed differential cell levels across tissues for the TC subtypes.
Yeah.
I'm curious if there's a difference in the rate at which those TCs proliferate in that spike time as a function of subtype.
Oh, yeah. Yeah. So that's a great question. That's work that we're literally just doing at the moment, where we're doing that sequencing data came from this mapping experiment where we were effectively taking mice at all of the different time points, and now knowing that there are different subsets, now characterizing the wave of TC1, 2, 3, and 4. And I haven't seen the finished part of that yet, because we're sort of collecting all the different time points, but I think that that's a great question. Do they all spike at the same time, or is it the TC4s that have this microbiota kind of related spike, and then the other ones will have a slightly different kinetics? But yeah, I think that's a really interesting point.
Yeah, so what are your thoughts on the breast milk versus the formula? Obviously, that doesn't apply to mice, but it does apply to humans.
Yeah, and I get asked this question a lot, and this is kind of the period at which you would probably want to exit a neonatal unit when you start this debate, because you don't want to get caught in the crossfire. I mean, the truth is that, and when I put up that diagram kind of showing breast milk, what I really want to say is that I think breast milk is going to turn out to be important, but it's going to turn out to be a modulator rather than an instructor, which is, it's quite clear from all of the epidemiological studies that we now have done in humans that yes, breast milk is important, but actually it's not that if you don't get breast milk, your immune system collapses, or you develop autoimmunity, or you don't develop tolerance. So, clearly I think breast milk layers on top of, a whole host of other cell intrinsic and extrinsic factors, which will regulate immune cell development. And the actual contribution from breast milk will probably, especially in humans, turn out to be kind of small, but it may not be unimportant, but it's going to be small. And I think that that's how we see it within the TC development. So we have a lot of this project now, which the students hate me for, because it involves removing the pups from the lactating mothers and then giving formula milk instead. And so you have to do this every 4 hours to kind of see what happens if you withdraw kind of breast milk, but you maintain nutrition. And I would say that our initial results are that the effect is modest, that there is an effect, but that it's small. And so the question is then how much does it matter if you, for example, have reduced number of TCs, but they're still there, but they're just operating at a reduced level. And that may come back to, what we see in terms of normal levels of variance from one healthy human individual to another. And of course, that in the end ultimately ends up setting your risk of certain diseases, but it's only one factor amongst many others.
So follow up. So do you see a link between the individuals fed breast milk and inflammation later in life, or is there data at all on that?
So it varies from disease to disease. So for example, the best evidence I think really comes from atopic disease and the development of things like asthma but the effect sizes are still very, very modest. And often what people show is that effectively, although you can find that there's differences in the microbiota, effectively that everyone sort of catches up to some extent, so that you may have initial effects during the first year of life where you see differences, but then the microbiota sort of normalizes. It's also the same, for example, for when you compare vaginal delivery versus C-section. It's like you can find differences as you track kind of offspring, but then they will eventually normalize. And so the question is probably, does it end up mattering for those that already had increased susceptibility to things like asthma? And maybe those are the ones where it really matters, but not that it matters for everybody.
I actually have a quick follow-on to that, but I do have another question. Does this imply that we could perhaps engineer a better formula?
Yes, so I think that is an interesting question. And then that's what I think we're really sort of thinking of it, which is, can you improve immune fitness during the first period of development, which is if we can identify what it is that is upstream that could give you high numbers of TCs during this early life period, exactly that. Could you imagine some supplementation approach where you could say, actually, yes, we could boost your TCs just during this period, but that would kind of set your immune tone, as it were. And I think there is actually a very interesting sort of mouse model of this, which is one of the papers I sort of cited at the beginning, where they did this very interesting experiment. So there are two strains of mice, which we commonly use, B6 and BALB/C mice. And interestingly, they have different levels of peripheral Treg cells. And that actually matches with their susceptibility to inflammatory disease if you try and induce like colitis in adulthood. And they did some very interesting experiments where they cross-fostered the pups between the two strains. And only if you cross-fostered them during the first few days of life, you could effectively switch the phenotypes. And so that suggests that there is something within the breast milk that can kind of set your levels. But it's not that, it comes back to this point, which is it's not like one mouse strain is missing peripheral Treg cells. It's just that they have slightly different set points. And so I think that what you're suggesting is absolutely that can you just alter your immunological set point in early life so that you have this sort of the better setup for protecting against inflammatory diseases later?
Just follow up on the same idea. Are we talking sort of being at the cusp of eliminating all possible allergies?
I mean,
I would love to have gotten rid of ragweed before I got too old.
Yeah, but there's a very interesting, I mean, obviously that is, for example, when you look at the data from the peanut allergy study, that is so striking, for the early introduction of peanuts, that I think that is the most compelling data that intervention does work. And these quite simple interventions in terms of just modulating when you're exposed to the antigen were clearly key. If you can also, as you said, alter when you get exposed to the antigen and make sure at the same time you're exposing them to the antigen that you also boost the same cell type that we think is going to be inducing tolerance to that antigen, you could imagine that you would make that intervention even more effective.
Make sure your kids are touching grass.
Yeah. Yeah.
Roughly, so the weaning period in mice was 2 to 3 weeks.
Yeah.
Roughly, what would that translate to?
Into humans, yeah.
Yeah.
So in humans, I mean, the current guidance is that most people wean their infants from milk to solid food around kind of 5 to 6 months. Obviously, for some children it's later, for some it's earlier, but that's kind of the window, which I think is pretty standard. And that's also interesting because that's also, if you look at Donna Farber's studies, where she has now started doing these immune phenotyping studies using these human samples that she's got through the pediatric transplant organizations, she would effectively endorse that, which is that pretty much she would show that your big transitions within your immune cell populations in the pediatric setting happen within the first few months of life. And by about 6 months, you've basically got adult phenotypes. And so, of course, she hasn't explored the antigen-presenting cell aspect, but certainly on the effector T cell side, and these are the cells that are being instructed by the antigen-presenting cells, it seems like the first 3 months is the really critical part. And, then by 6 months, you're kind of at your adult levels.
Do you know if steroids after, like, right after birth to
Yeah.
a premature baby, if that affects this outcome?
Yeah, I don't know. I think that's an interesting question now, which is for us, sort of how much of what happens during early life impacts on immune cell development. So exactly as you say, a lot of the things that happen during the neonatal period, now we have this ability to obviously to intervene very early and support preterm babies, but that comes with a whole host of exposure to a number of medications. Obviously, antibiotics is like a key one in terms of the number of courses of antibiotics that are given on the neonatal unit. And that's a kind of key area of interest. What happens if you disturb the natural order of progression in terms of microbial colonization, and then you see microorganisms outside of these like key developmental windows, I think is also a great question.
some chemotherapy treatments often have a side effect of basically the patients who survive end up with new allergy.
Yeah.
So is this possibly the mechanism by which that occurs? They lose their long-standing like tolerance, food tolerance for whatever dietary allergens there might be.
Yeah, it's a really interesting question, and we think about this also a lot in the context of bone marrow transplantation. What happens if you go in and you basically give someone a new immune system, but outside of the normal developmental window for when you were supposed to encounter a lot of these environmental antigens that you then are supposed to develop tolerance to. And sort of related to your chemotherapy question, one of the very first research projects I ever did when I was a medical doctor was I spent some time in a lab with an immunologist who specialized in food allergies. And we were actually investigating this very interesting phenomenon, which is not dissimilar from what you're describing, which was what happens to children who go on to develop liver transplants. And interestingly, what they had shown is that children who develop severe liver disease actually have a very high incidence of oral allergy. And that's perhaps not surprising because they have an inflamed liver. You can imagine that they also have disturbances in these tolerogenic cells that should be inducing food tolerance. And that doesn't resolve, obviously, when you go on to give a food transplant, precisely because you're probably then giving the transplant and at a stage where it's too late for developing kind of tolerance outside of the normal windows. So I think it's not dissimilar from the phenomenon that you're describing with chemotherapy, which is you can almost be resetting immune cell development, but outside these normal cues for when these cells would be present. Perfect.
That's it. I want to again thank Dr. Brown for coming and thank the Dean's office and thank you, Rise, for sponsoring her visit.
Thank you. Thank you very much.
Invited back to the atrium in Science Hall for a reception to ask more questions that you may have. Maybe ask some questions, some more personal.
Say a little bit about her career journey, or are we out of time?
What time is it? Do we have? Will you give us 3 minutes?
I'll give you a 3 minute too, and then anyone who wants to ask me questions afterwards, please feel free to jump in. So if you can't tell from my accent, I'm from the UK. I was born in Cambridge. I went to university at Oxford to study medicine at New College. And then after that, I moved to London, to University College London, to do my clinical training. And then I started training in pediatrics, and then later started my PhD with an immunologist called Randy Noel. Randy Noel was actually from Dartmouth in New Hampshire, and he kept his lab in Dartmouth. And I was saying over lunchtime, he used to get extremely frustrated with how long it would take to get approvals to do any kind of experiments in the UK, particularly animal experiments. And so what used to happen was, as soon as he wanted us to do something, this would happen. And we would have to fly over to the lab in Dartmouth to spend a few months there, finish your experiment, and then come back. So I had this incredible experience of being exposed to science within a big lab in the US in Dartmouth. And I think for me, that was really where I got the bug for research and knew that this was something I wanted to pursue as my career. And so when I had the opportunity to do a postdoc, I was looking back to the US and thinking, where could I find an environment like that? And so I moved to Sloan Kettering to Sasha Rudensky's lab for my postdoc, and had 4 fabulous years there. And so after that, I was thinking, well, I always thought I would go back to the UK and set up my lab there. But actually, it was just too hard to leave because science in the US, I think, is so fantastic, particularly for students. And it was actually talking to the students in the UK versus talking to the students in the US that made me realize, actually, if I was going to build a lab, I'd want to build it a lab with students from the US. So then I set up my lab here at Sloan Kettering and within the Weill Cornell immunology program. And that's where I've been for the past almost 3 years. So I'm very happy to take more questions from students over the drinks.
Again, I invite everyone back to the atrium in Science Hall. Yeah.